Hi everyone, I am Anam Javed. I'm an undergrad final year student at Amity University, Noida, India. I'm a research fellow at Pine Biotech, and the project I'm currently working on is identification of biomarkers leading to Alzheimer's based on RNA sequence of autistic and control cortex. So, Alzheimer's disease is a disease which affects mostly the brain and causes a symptom called dementia. It is said to be progressive cerebrum disorder, which is irreversible and that gradually destroys thinking ability and old memories. It is said to be a disorder which is irreversible and that gradually destroys thinking ability and old memories. In this picture, you can see how a healthy brain gets affected by Alzheimer's disease. Alzheimer's disease and autistic spectrum disorder. Autism spectrum disorder and Alzheimer's disease are neurodevelopmental and neurodegenerative disorders, respectively, with devastating effects. It is of utmost curiosity that these disorders express at different stages of life, and there is an involvement of certain susceptible genes. This genetic basis makes the background of common associations like memory deficits, cognition changes, demyelination, oxidative stress, and inflammation, an integral part of both disorders. The methodology which I used was a pipeline was run using the dataset which was RNA sequence of autistic control cortex. A comparison was made between the autistic and control cortex genes. Comparative analysis done to analyze the sample between the groups by running the expression pipeline on tBioInfo server. As a result, DSEC2 was used to obtain differential express genes. Enrichment analysis is used to obtain KEGG pathways. GSEA functional enrichment analysis was used to obtain geo-enriched pathways and the network. Comparative analysis. Comparative analysis consists of pipelines to obtain differentially expressed genes, KEGG pathways, geo-enriched genes among the groups. DSEC2 is a tool used for differential gene expression analysis of RNA sequence data. Enrichment analysis identifies biological pathways, that is, KEGG pathways. GSEA analysis is done to determine set of genes which show statistically significant concordant differences between two biological states, that is, GO pathway. Like here we can see how the pipeline was run. First we click on the start button, and then DSEC2 is selected, then enrichment analysis and GSEA according to the requirement, and then we end the pipeline. The parameters are fed into the pipeline. Volcano plot. It is performed under the two conditions, the adjusted p-value at y-axis and log2 fold change at x-axis. It determines significant differentially expressed genes. Genes which are having negative log2 fold change value will be considered as downregulated genes, whereas the positive log2 fold change value to be considered as upregulated genes. It basically determines significant differentially expressed genes. The blue, the blue area represents significant genes, and red area represents significant differentially expressed genes. Genes which are having negative log to fold change value will be considered as downregulated value, whereas positive log to fold change value as upregulated value. Basically, the higher gene in the plot is more significance. KEGG enriched pathways. KEGG provides information of enrichment of genes in the area displayed on the basis of two parameters, count and p-adjusted value, which shows distribution of genes. As you can see here, that count shows the number of genes involved in the pathway and color represents the adjusted value. KEGG enriched pathways provides information about various pathways that are either activated or suppressed due to the upregulation or downregulation of the gene respectively. Pathways corresponding to the highest quantity of upregulated genes are highly activated, and pathways corresponding to a higher number downregulated genes are highly suppressed. So basically, the highest count of upregulated genes is associated with cytokine-cytokine interactions. As you can see here, it has the highest count number, and the downregulated gene is associated with TCA or citrate cycle, as we can see it over here. Network plot. It is basically a graph that analyzes the pathways in the network based on the fold change value. Size of dot in the code depicts the number of genes involved in particular pathway. The color indicates the fold change value. It analyzes the genes that are included in the particular pathway. The dot size depicts the number of genes involved. Color of the dot fold change value shows the fold change value, and decrease in the fold change leads to increase in gene expressed in pathway. As we can see that there are a few core hub genes, which over here are is NLRP9, and over here it is SOX4, CNMD, and TSPO. So the key findings which I have noted from KEGG analysis is possible association can be made between suppressed pathway of citrate cycle or TSA and cytokine-cytokine receptor interaction. From Volcano plot, the upregulated genes is LILRB1 and SOCS3. Also, we found hub genes which can be used as biomarker in network plot, which are NLRP9, ELF4, TSPO, CNMD. Future directions. Researchers can perform experiments focusing on these particular gene found in the study and correlate their upregulated and downregulated genes with disease progression. Thank you so much.